And we're gonna continue now with the overlap correction part, which is also called infinite response modeling. So far, what we've been discussing was, well, good old ERP event structure, right? So what we had was basically you show a stimulus, or we treat everything as if everything is just separated in time, right? Everything is far away. But it turns out that often in many paradigms, especially if you go for more natural environments, everything is happening at the same time, right? So for instance here, what I showed you here from this nice, from the paper with Olaf together, we had a simple design where somebody's looking at the spikes or the faces. And if you can imagine for a second that each of their eye movements elicits an ERP, right? Then these ERPs, they typically go on for longer than the 200 milliseconds that we on average have time until the next eye movement appears, right? So they are overlapping in time with each other. Of course, this is not what this is, if you would know exactly each individual separate one, that would be perfect, but you don't have access to that. You only have access to this overlapping one. And here in this animation that you probably had time to look at, I make the overlap smaller and then I make it larger again. So very similar to this first notebook that you were working on, where there was this minimal overlap, and you could also include the overlap back, add overlap to your data set. And now you can say, okay, so what do I care? I can leave this meeting now because I don't do eye movement research. But this is not only for eye movement researchers, and I will show you a very stark example later. Worked with Romy together. But this is also can happen if you have a very simple P300 experiment where you ask people to do button presses. And probably even you have it in there if you don't ask them to do button presses, but then you don't have an event. So you can have an event onset from your condition, whatever that is, target, non-target, and then you have a response, a button press response. You will typically measure that in the same electrode because everything is measured in everything. And if you, let's say, are faster for targets than for non-targets, which you are typically, then you would get a systematic bias in overlap between these two ERPs, basically. And so, yeah, you should better take care of that. Okay, how do we do this? I have here, I just take this example once more. This is what you're measuring. This would be an ERP without taking anything into account. And I'm now including also effects of covariance. And of course, that would be great, right? Now you have a difference between bikes and faces and all these different places. You can publish 4 papers on that. Perfect. But we would like to know what are the underlying ones. This is just the good old ERP. To drive this point home of why this can be a problem, let's have a look at this animation. If you have no overlap like you're here, then obviously what you're measuring has the same shape as this curve that I simulated without overlap in the first place. If you now add overlap, then this overall ERP shape will differ because you will have before and afterwards, you will have other of these shapes adding activity to that. And depending on the shape of your overlap distribution, this might look different. So now imagine this is your fixation duration or your reaction time, doesn't matter. And you have these 2 conditions and you have a difference between these 2. What that means is that you're basically sampling this orange line at 2 different places, something like this, right? So you have this difference of overlap distributions. And so then overlap artifact starts to crop in because it takes for the bike fixations, it takes a longer time. Like there are less bike fixations or less other fixations back here than for the face fixation, they are closer by. So you have a systematic difference in overlap. This is actually exactly the same effect of why you should also take care of your nonlinear or effects, because here, let's say you have a nonlinear effect on the P1. You also have a distribution of your predictor. And you have a difference between these 2, which also means that I'm sampling maybe the one condition I'm sampling here and the other one I'm sampling here. So this would be a difference that is just solely there because I'm looking at the data at 2 on average at 2 different saccade amplitudes in this case, right? So summarizing this was our simulation. Maybe we even have a true effect that we are adding now. We had this biased overlap and biased covariate things. So we have all kinds of effects. This is what we saw before. If we fix the overlap, and we will see in a second how we do it, then already this looks much cleaner before and in the end. And if you also fix the covariate, because we include it and we do a marginal effect at the end, then also this effect goes away. And this is, of course, simulated data. So yeah, but you will see in a second how close we are actually to the true data as far as we can tell so far. Now, this one we know already. How does it work? But we have no idea how to do the overlap. And also, it's maybe at first sight, it might be very surprising that you can even do that. Like, what's the magic behind it? What additional feature can we use from the data to actually disentangle all this mess and go from something like here, overlapping, to something like here where we have the individual ones? And I think the key insight you have to have is that what does it mean to have activity here, right? This activity here has, of course, a certain distance to an event, right? And if we think of this hypothetical non-measurable space here, then we know that this event probably or might have activity that will be measurable in the observed, in the overlapping EEG. This activity also can come from a different event further back. And then we know, because Maxwell's equation, etc., everything adds up linearly. So if there's no interactions between the events, let's assume that for now, then we could just say, okay, this, what we observe here, is just the sum of all the activity of all the previous events, but importantly, at different time points. So this is the response after P1, N1. This is somewhere the response in the P3. But here, this is the response in the N1. Right? It is the same event type, so we will have more or less the same shape, except maybe saccade amplitude, nonlinear effects, blah, blah. But thereby we have this. Okay, how does that help us? Now we know, okay, this point is probably the sum of two responses of the brain. We can just assume that. But we don't know the shapes, so we don't know where to put activity to the one or the other event. Luckily, we can find exactly the same activity here in the next event as well. And now here, if you look here, this is a sum of three different events, right? I'm ignoring events that are super far away back. So yeah. So this is a sum of many more events. So here we have this data point, which is a sum of two events, basically. This data point is a sum of two events at different spacings. And that's exactly, because we can do that for all our events, this is exactly where the variability comes in, that overall, if we put up a system of equation, then we can try to pull these things apart and find the solution that best explains the observed data, given that we know the distances of these to the individual events. Now going back and bringing this together with what we learned earlier with the linear model, with the design matrix, etc. So also, why do I put so much focus on the design matrix? Because it will be so helpful to understand this part here. We're doing something slightly different. Before, we had single trials here, epoch data. This is like the thing that you always use in EEG's ERP analyses. But we are going away from that. We have continuous EEG, and this is important. This is really a conceptual difference. We are not actively epoching the data here. We are epoching the data in the linear model, right? So this is continuous EEG data from sample 21 to sample N, right? And next, we're going to build a very, very large design matrix. Instead of fitting one time point, now we have to fit all the time points at the same time, right? And that means multiple things. First of all, an intercept is not only one column, but suddenly it's one column per time point of your, let's say, epoch, your local time epoch. And the continuous effect would also be not only one column, but it would be multiple columns. In this case, it's only 5. It would be a very short EEG, but this is, of course, an illustration. You would have 500 of those. You would have 500 predictors for, let's say, 500 samples in a 1,000 hertz data set. And not only your columns are many, but the columns always match the betas. So here your betas are also many. So here I have 10 betas fitted at the same time. I colored them because one belongs to the continuous slope and one belongs to the intercept here, basically. But we are fitting all of these at the same time. In the end, what we're really doing is we are trying to predict the continuous EEG, which I think is a very ambitious goal to have. And how far we get there, well, we don't know. And of course, we have some residuals because we will not be able to explain all of the brain activity just by a few events that we are specifying in our model. Okay, now the important bit is now what happens in the design matrix. And you get this kind of staircase, we call it staircase function. For people who do fMRI, this is FIR deconvolution. This is nothing new, has been done since the 90s, but maybe even nonetheless interesting. So what happens here? Here we have an event. So we put in, this is this sample here should be explained by this event immediately after the onset. The next column, we shift this by 1, because now this one here should be explained by the event activity that is 1 sample distance from this. And here 3 samples, etc. And the same is here. They are in parallel because this is the continuous value that belongs to this event here. This event here down here has a different continuous value because maybe it's the next maybe it's the next saccade or so, different saccade size. Now there's no overlap, right? We have a certain time window, 5 samples that we're modeling, and we're assuming everything outside of this time window, there's no activity anymore. But what happens now if I push this event closer, right? I can push this closer. Then the design matrix changes, and suddenly 1 sample is not explained only by an intercept and the continuous predictor, but by this intercept here and by this intercept here again, because now we are in this domain where this first event still explains the sample, but this event already also explains the sample. So this is how we get the overlap correction in. And because at a later stage, this distance between the events will be different, we will always have a different combination of events, and that's what allows us to solve this problem. Yeah. Nevertheless, okay. That is the explanation of why all of this works. There are some technicalities behind it, because as you can see from all these dots, these are all zeros. So this is a very sparse matrix that we have to do. And if you think of how long an EEG signal is, it can be 3 million data points. And then I already mentioned here, let's say 500 predictors per, like 500 columns per effect here, and you have 5 effects. Then suddenly you have 5,000 predictors to estimate and 3 million data points. And that is, of course, where Unfold really shines, and where it has its special solvers to address exactly that problem. But all of this works under the hood. There are some tricks that we are nowadays doing that is more for advanced people, where we can put this stuff on the GPU, where we can do other transformations, etc. And we can really trim down the fitting time of these models by, compared to the MATLAB version, a factor of 100. It depends a little bit on the specifics. But yeah, it's really nice to be able to fit a very complex Unfold model in less, 400 channels in less than 2 minutes or something like that. Yeah, there's more on the Unfold website if you're interested in that. How does this now look in the wild, right? So this is a toy example, of course. In the wild, the design matrix would look like something like this. So you have, again, I plotted the same way. So you have time here, and then you always have the staircases depending on, here we have yeah, this is just also some kind of simulation with a button press, maybe a button press event, maybe a phase stimulus event. I forgot what the intercept is. Okay, but it doesn't matter. Now you could rightfully say, okay, this is a nice story and it's a nice idea, and it looks pretty, and you can give talks about this, but does that actually make any sense? Does it work? Is there no interaction between this kind of saccades? Like, what can you show me that might convince me that that this is actually something that makes sense? And like, for me, this is the I'm so happy this turned out exactly like that, because now you saw the simulation. Now let's go for real data. This is work from Gert, and she was interested in free viewing of face processing, faces versus other objects if you just look at stimuli. And this is the non-unfold, the classical ERP plot, right? And as you can see, yeah, you do get the N170 phase difference, what you'd expect, but there are many other differences that you can see here. Right. And so we were ultimately, in the beginning, we were super happy because we thought like, oh, maybe this is some kind of preview effect. You see the face in the periphery and that's why it changes, etc., blah, blah. But, well, yeah, it turns out otherwise. So if we add the magic of Unfold, pling, right, then suddenly the baseline and the ends, all these differences, they completely go away. And the only thing that remains is the N170 phase effect. And for me, that is I mean, if you're a researcher who's interested in whether the lab experiments generalize to the real world, which everybody should be, then this is kind of a dream result, because this is exactly like in a more ecological valid situation where you have eye movements, you get exactly the same result. How awesome is that? Why was now there? Where does this overlap come from? And mostly the reason why this happened was that people, if you show them an image, they will first look at all the faces and then they will look at the other objects. So there was an overlap, not only a problem between fixations, but also with the stimulus, a very strong bias there. And that explains this early thing. This later, here's more the different eye movements. Okay. I give you another example. I give you 3 examples in total. This is also something, yeah, well, Olaf is there again. This is a study he did some years ago in reading, right? So it's a reading paradigm. You read a sentence, in jedem Haushalt findet man dort. And then there's a keyword here with an invisible boundary. So if you focus your eyes here, in one condition, you would have a valid preview. In another condition, you have like a jumbled preview. And that's exactly the question. What's the difference if you have a valid preview versus non-valid preview? Can you use the peripheral information to help you read a sentence? And once you landed online, the eye tracker measures once you landed on a word, always the word will be displayed. And we are looking at this fixation here. So you're actually fixating the word, and the 2 conditions are just whether you, in the periphery, you saw a correct or an incorrect preview. So valid or invalid preview. And what people generally found since many years is that if you had an invalid preview, then you were your fixation duration on that word was slower, right? So it was longer. So if you look at the ERP of this effect, then you will have a systematic bias of when the next fixation comes in. Okay, so we have a systematic overlap bias in this experiment. This is what the data look like without Unfold. So you have some kind of ERP, and then you have, after 200 something milliseconds, we get 3 different clusters of differences. This is the difference effect here, different curve. And here, this is the ERP image. I don't have too much time, so I will not. If you don't know what an ERP image is, yeah, the trials are sorted by when the next fixation comes in. Right. So now if we do use Unfold, then it looks slightly different. So this middle effect kind of went away. You can also see it in the difference curve. And in the ERP image, this next fixation, this is what we are basically were able to remove. And now we could also only look at this, what we are removing. And if we do this, then we can see that this is that we actually see exactly this systematic overlap difference. We can see it in the only overlapping activity, and this is actually significantly different from each other. This is kind of the difference between these 2 as well, right? So given this is also, for me, seems to be a very sane way how to empirically test this idea. And now for the latest one, this is a study we did together with Romy, and this is, I think, where we steered the bee's nest most strikingly. And there's ongoing discussion whether our analysis is even valid or not. I'm very happy to discuss it here as well. There are some issues. Okay, I can't go into the details. It's a little bit complicated, but I do think it is quite compelling. So what Romy is interested in is decision making. So why do you choose to eat a burrito or eat the chicken during lunch? These kind of questions. And what people have been done in perceptual learning is that they have been using this kind of drift diffusion models, where you assume, oh, you're integrating information for either of your options, and at some point you reach a threshold, and then you're going to do your response. So they were also looking into EG, and at the CPP, they found the CPP effect where for easy decisions or fast decisions, you get this faster ramp up than for slow decisions. And here, this is 4 different data sets. This is kind of the effects that they were looking for. Sometimes they look a little bit more that they reach the same point, but there's a lot of theory behind this, why this does not necessarily have to be the case. And if you do the difference, there's always a difference between having these kind of things. But now, one issue that they have sometimes try to address, but we argue that they have not fully addressed, is that if you have an easy task then your reaction time will on average be faster. So what that means is that your response and your stimulus will also be closer together. So there will be a lot of overlap between these two ERPs. If you have a hard task, then the reaction time is slow, and then also these two things will be further away, so you will have less overlap for these two things, right? For the ERP, if you look at the ERP like this, time log to the response, for instance. So, well, basically, what Romy figured out, if you apply Unfold to this, then these differences, they just vanish, right? We find no significant difference anymore. Maybe there's a little bit something like here. Or at least we can definitely say there's very much a reduction of this kind of effect. This application of Unfold is a little bit criticized, and it's a little bit complicated why or why not this is the case. To make a story short, it's if you have a perfect drift diffusion model there's no jitter between the stimulus and the onset of the ramp and the offset of the ramp, then you would not know, Unfold wouldn't know whether to apply, whether to put this activity to the stimulus or to the response. So in a very clinical situation, this Unfold would return a little bit nonsense results, or depends on the parameters, can return things. In the real world, we argue that this is not so much a problem because there will always be a little bit of jitter and it will always be tuned more to one or to the other one. But yeah, okay. This is definitely a very striking result and something to be explained for this community as well. We would have been actually very happy if this is not the case. Or I think Romy I mean, I'm happy that this is the case so I could work together with Romy. But I think Romy would have just loved to continue on with the drift diffusion models like that. It would be a very nice signal to have in the brain, I think. Okay, anyway, these were the three examples I wanted to give you to try to convince you that there is something that you're modeling that is correct, and at least maybe convince you that the lower thing that you see here is a more realistic version of what's going on in the brain than the upper version, which is definitely biased and to some extent, right, from first principles. Okay. Now, how do we do this in Unfold? In Unfold, now we have to, we slightly shift gears. We have to define one more thing. So we had the data before was channel times time points times epochs. Now, you remember, we have to simulate we have to analyze the continuous EEG. So now we have just channel times time or channel times samples. One big matrix. We also still have the events. Now, importantly, we need this column latency. We need to know when an event is happening in your continuous data. So the latency is always in samples, like in EEG Lab. We still need a formula, and we can define different formulas for different events. We can even define different time windows for events. You will also learn that, but let's not jump ahead. And now we also need to tell Unfold what kind of staircase to use. And this is, I think, most users will just use the FIR basis. You need to provide it a time window. This is a very common question of how long to put it. This is basically what you're, if you're epoching the data, you also have to put a time window. This is basically that time window. You want to capture all the data of this, all the activity of that event. We have a paper on selecting like how bad it is to make it shorter, and it's not that bad. But yeah, I typically recommend just to have the whole thing there. And you have to provide a sampling rate so that it can translate these kind of numbers to samples, because all the Unfold toolbox pretty much is in samples, except some of the input functions. For convenience, we allow to place some seconds or milliseconds, whatever, here seconds in this case. Then what changes is that instead of the times vector, we now put the basis vector here in the end, and that's it. And I found a mistake here. This should not be data epochs, but this should be data. Sorry for that. Okay. But yeah, you get the point. And in the exercise, of course, you get some hints of how to do it. After you have the model, actually, everything is pretty much the same. You can use CoRFM and you can use CoRF table. Here you will get, you will get slightly different output because now all the betas are attached next to each other. We cannot build this nice matrix that we had before because it could be that the different predictors, the different events have different basis functions with different lengths. So yeah, some of your beta strings will be long, others short, etc. Yeah, anyway, you will see that's why I really like this kind of tidy data frames. Yeah, and I think with this, you are ready to start exercise number 4. Questions. Wait, I will also look at questions. Okay, okay. Yeah, I will, to Tom's question, whether the window length need to be the same for every regressor. Okay, so it needs to be the same for every event. I think that only makes sense that if you have an intercept and a continuous variable or so, that they have the same length. But then you will see we have a multi-event exercise also. You can define that the stimulus should have a very long time window after onset and very short baseline, 200 milliseconds or so. But the fixation should look more symmetric, or a button press should even look more to the back. So they can absolutely, different events, I think conceptually make sense that they actually have different window lengths. Yeah. There are 2 more things that I briefly wanted to discuss. One pertains a little bit of what are the assumptions or the things or the limitations. And René is still figuring out the limitations in a project called Break Unfold. But one definite limitation is that if you have no or very, very little jitter between your events in your data set, so the events are in each trial, it's exactly 400 milliseconds distance, then there's no real way to separate these two. You never will know whether the first event or the activity in the middle is actually relating to the first event or relating to the second event, but previous in time, right? So you could assign it to both of them. So you need some kind of jitter in your data for overlap correction to work. I think this is the first point. I think that's important to keep in mind. And the second one is that if you want to have single subject significance values based on this, then this is a little bit tricky. And this is also tricky in fMRI. That's why they use all this kind of whitening and autocorrelation function or so. And the reason is that if you have this one large, big model, that all the residuals, they have a correlation with each other. So you cannot, like, if you would ask the linear model, hey, how many trials did you see? Then it will not know anything about how many events are put in, but it will only know about how many samples it has. And then it says, awesome, I had 3 million trials, right? And all your standard errors will be tiny, tiny, tiny small. Right, this is not the whole truth. Actually, Felix, who's also here today, he did his master thesis on such kind of effect, and we found very mixed patterns. But now I think with Unfold Sim, we should really revisit that. So Felix, if you're up to it, we can do a small simulation study again, repeating your master's thesis now with different tools. But yeah, this can be a problem to find single subject significance values. Yeah, I wanted just for clarity, I want to add that.